I'd like to welcome everybody to the March 17th meeting of the Modeling and Viral Pandemics Working Group. We have two speakers lined up for today who've been very generous with their time. They both agreed to stay and have a conversation after their talks if there is interest. Appreciate that very much. I need to remind you that this meeting is recorded, live streamed, and publicly available. As always, Reinhard and myself are available to take your suggestions, critiques, ideas. Jim Sluka and Lorenzo also are available to help and to think about problems. We very much appreciate your feedback and suggestions. We have a variety of communication channels. I think people are familiar with these by now. We have a Slack, a Twitter account, a wiki page. The wiki page especially could use some love, a LinkedIn group, and our YouTube channel. We very much would appreciate help in disseminating this material, of which there is a lot, and making sure that people are aware of our projects. So please do help us disseminate that material. In particular, we have the Twitter feed, MSM Viral. If nothing else, we put our weekly seminar announcements there. But please do help us retweet that and ensure that we get an appropriate audience for our speakers. Wanted to ask for announcements. As always, we're interested in any products that have a connection to the working group. I wanted to say that out of contacts through the working group, we have just published a paper called Multiscale Modeling of Antiviral Timing, Potency, and Heterogeneity Effects on an Epithelial Tissue Patch infected by SARS-CoV-2. Giuliano, the first author, has spoken to this group. Tarun and TJ as well. Jim Sluka, as you recognize, is an organizer. Sarah Quinney is in our School of Medicine. Morgan Craig, you've heard speak in the context of Paul Macklin's modeling work, and Robert Stratford has also spoken to us. So that's a recent paper that grew out of this working group, and we would appreciate it if there are any papers, projects that you develop through the working group that you would let us know and help us publicize. We have a steering committee meeting tomorrow morning at 11 a.m. If you think you should be invited to that steering committee meeting and you don't have a link, let us know and we will provide the link to you. Are there any other announcements before we continue? Okay, hearing no announcements at the moment. You're always free to talk later if you like. We'll continue with our first speaker. Before that, our schedule for upcoming meetings and mini seminars. Next week, we have one talk scheduled at the moment. Coming up on April 7th, we have a talk on modeling blood clots. It sounds quite interesting. For the first time since this series began, we have a week where we don't currently have anyone scheduled. So if you have any ideas for speakers for March 31st, that could be somebody from a subgroup, a discussion forum, or anything else, please let us know. Lorenzo is extremely active in securing speakers and sending out invitations. So if there's anybody in your group you would like to have speak, graduate student or postdoc, you'd like to speak again, or if there's somebody you'd like to hear, please let us know, and Lorenzo will do his very best to secure them for the series. So we always appreciate your input and help in coming up with speakers and ideas. Since we have 2 speakers today, we will do our usual approach. We'll have the 2 short talks followed by questions. If people can remember to mute their mics and hold their questions until after the presentations, that would be great. If people have an urgent question after a single presentation, we'll do our best to accommodate that. Again, I'm on a laptop today rather than my usual computer, and so it may take a little bit more effort for me to see you. If you need to ask questions, feel free to do so. Our first talk will be on crowdsourcing and benchmarking to understand viral susceptibility, and I will turn it over to our speaker.
So I'm going to talk about crowdsourcing and benchmarking, also known as data analysis challenges. And I'll talk about it generally, but time permitting, I'll describe a challenge that we ran on viral susceptibility, since I think that's what this crowd is most interested in. So I work for Sage Bionetworks, and for those of you who are not familiar, we're a small nonprofit out of Seattle, Washington, and our mission is to speed up biomedical research by sharing data, by enabling collaborations, and data analysis challenges are just a different kind of collaboration from our perspective. So why are challenges important? Well, because self-assessment of modeling leads to what we call the Lake Wobegon effect, where everyone is above average, right? So in order to get your model published, you have to compare it with other models. And there are a surprising number that seem to beat all of the other models, not necessarily because they would beat them in every sort of metric, but because people tend to paint their models in the best light. So some sources of heterogeneity that lead to the self-evaluation pitfall are the data used, or even if you're using the same data, the version, the QC, the pre-processing, and then there are modeling specific effects like training test splits, choice of evaluation metric. And then, as much as we all think that we don't, there's inadvertent data leakage. So benefits of challenges. The first is benchmarking. So it's really a way to compare head-to-head, apples to apples, different models. It can accelerate research by crowdsourcing a large number of people to work on important problems. It helps sample the space of models so that you can understand what models work best in the given scenario. And it's also community building. So we've had a number of collaborations come out of challenges that we've put on, and more and more we, as a feature of the challenges that we run, we include a community phase. So, people compete at the beginning, and then we gather competitors to work together to understand the results. Crowdsourcing is not new. The first identified instance of crowdsourcing was in 1714, where the British government challenged people to build a device to measure longitude. So while out at sea, it was difficult to measure longitude, and so the first known crowdsourcing event was to solve that problem. Another well-known source of crowdsourcing was the Oxford English Dictionary, which had the first 1884 version had over 800 contributors. And now today there are a lot of different crowdsourcing mechanisms, from Kaggle and Incentive, X Prize, Netflix Prize are all kind of the famous ones. But even things like Waze that crowdsource, information about traffic accidents and speed traps and things like that. In the space of computational biology, there are actually a number of players, of which DREAM is the one that SAGE is involved in. So as a community, we've had over 30,000 solvers worldwide. I think of the challenges I've been involved in, the most exotic location was Fiji, but our solvers come from all over the world. And at this point, we've run over 60 challenges. A DREAM challenge requires a lot of work on the front end by the organizers to assemble the data, to identify the metrics that'll be used for scoring, to make sure that data can be shared. But this culminates in what we call an open phase. Like I said, we've had over 30,000 solvers to date in over 60 challenges. Once they submit their models, we have an analysis phase, and more and more recently, a collaborative phase where we invite the participants to help us do a post hoc analysis of the models. Out of this, we've had over 100 journal articles, some special issues. We've supported 20 plus PhD theses. And we find also that professors like to use challenges in the classroom. So we often get groups of students competing. We've also had a few instances where commercial entities have spun out of challenge results. So most challenges are set up to answer a specific biological question in a specific context, but we also have examples of challenges to identify best general methods. So these are a set of challenges that were set up to benchmark methods for somatic mutation calling, both in DNA and RNA, and they also established the data sets that are often now used as benchmarks for these this task. We've also run some challenges that, well, not in DREAM, but at SAGE, we've also run some challenges that are more general, like ideation challenges. These are much more open-ended, where we share a data set, and instead of having a specific prediction task, it's a more general reuse of data that's then judged by a panel instead of using a machine learning metric. But most challenges run in this general way. We provide a training data set on which participants develop their algorithms. There's often a second validation, a validation set that against which they can test their models in an iterative fashion. This isn't the final data set, though. This is more for helping the development process, for kind of checking in to see where they are. But then final evaluation is done on a third data set, and then using the scoring metric, the predetermined scoring metric, we rank the final solutions. Oftentimes, and increasingly, there can be multiple phases. For instance, if there are more and more versions of test data or training data available. So this is the most general framework. It's been shown that in many cases, aggregation of results across teams result in models with better performance than the top performing models. Those of you who are familiar with ensemble modeling know this phenomenon, but that's another feature and value that comes out of crowdsourcing around sampling the space of models and then aggregating across them. So in the example or in the general framework I showed you, the data are made available to participants, but we can also operate when data are not actually available to participants. So this is an example of a challenge we ran several years ago to use mammogram information to diagnose breast cancer, essentially. And the data consisted of mammograms from the Group Health Organization, which is a health organization here in Seattle. Now, because these are actual patient data, they were not consented to be shared. And so we actually couldn't share these images. We couldn't, both because of the size of the data set and because of the sharing issues, the data couldn't be shared with participants. And so we developed what we call a model to data framework, in which the data are actually firewalled within our servers, and the participants work on their models and actually send them to our servers to be evaluated. So this is great, both when data are large and can't be transferred, and when data can't be shared. And since then, we've run a number of challenges using this framework, using patient health records and images, among other things. Yes, so the models get sent across the firewall to our servers to actually run on the data. Next, I'm going to describe a challenge that we ran in 2016 and 17 on susceptibility to respiratory viruses. This was motivated by the fact that gene expression signatures can predict which patients will get sick following exposure to respiratory viruses, but that signature prior to this challenge was evident really starting about 36 hours post-exposure. And so we wanted to know if we could identify early stage predictors of viral infection. So we collected data from 7 different human viral challenge experiments. Challenge is now in a different context. I think this crowd is probably much more familiar, is probably quite familiar with this setup, but basically you take healthy volunteers, you expose them to a virus, and you see which ones get sick, which ones don't. Meanwhile, they took blood samples at regular intervals throughout the course of infection. The endpoints that were looked at were symptom severity. So this was a summary of 8 different dimensions of respiratory viral symptoms. We also had viral shedding data. And then gene expression was pre-RNA-seq, was done on a gene expression array chip. like I said, we had 7 different studies that were sampled mostly at similar intervals, but there was some differences across the data sets. We had originally envisioned looking at this challenge by increasing time post-exposure. So the first set was pre-exposure, or basically up until viral exposure, 12 hours post-exposure, 24 hours, and 36 hours. We had 4 of the data sets we had available to us were previously publicly available. 3 of them were not, and so we pulled the test data set from those 3 data sets that had not been previously available. And we formulated 3 sub-challenges to predict viral shedding, to predict binary symptomatic response, and to predict continuous log symptom score. We had originally envisioned 4 phases corresponding to those different time points. So where we released increasing portions of the data. So phase 1 is that up to time 0. Phase 2 was up to time 12. Phase 3 was 24, and phase 4 was 36. Unfortunately, we had an inadvertent test release of the test data by someone who was not related to the challenge, but was a postdoc in a lab, and released the data along with one of their publications. So this actually turned out to be a really interesting benefit. We re-envisioned the challenge pretty quickly. We then focused only on the 0 and 24 hours, but we made this a much more collaborative envisioned effort, because we lost our original data set. We did test data set. We did find another one. And so we were able to test these models in an independent data set a little bit later. But in the meantime, we created we made a more collaborative effort where we asked for, in addition to the predictions that we normally do, we asked for predictor lists and leave one out cross-validations, which are used to build ensemble models. And code and write-up, we typically require code and write-up. And we additionally asked for permutations of the models. So in the end, this is, make sure I should probably skip to the punch to keep us on time. In the end, what had been the test data set became a leaderboard set, and we did eventually validate the data in an independent data set. One thing that's important to note is that those time 0 and time 24 time points were before the onset of peak symptoms. So this challenge is really about predicting viral exposure before people truly get fully sick. What we saw was that for symptoms, participants were able to predict using at both time 0 and time 24. They actually showed slightly better enrichment of their predictive models at hour 0 pre-exposure rather than time 24, which is surprising to me. And we saw the same thing in log symptom score and the continuous measure of symptoms. What was surprising was that for viral shedding, we didn't show an enrichment of prediction. So the viral shedding models were not significantly predictive. And that's likely due to some overfitting on the leaderboard samples relative to the final scoreboard. It's also important to note that the final scoring was in an RSV only data set. So the training data set was maybe not totally, well, there were RSV samples in the training data set. The general models did not translate into the RSV scoring data. So we did have an active community phase to answer some guided questions because we had lost our test data set. We had set up this community phase. We had a very active group. Everyone who participated was eligible to, everyone who submitted a model was eligible to participate in the community phase. Some of the questions we asked were, what can we learn about the biology, from the predictor set? We had a group looking at whether some subjects were inherently easier or harder to predict. We had one group that was looking at the permutations to see if model overfitting can be assessed via permutation in the absence of an independent test set. We had one group that looked at the set of approaches that read all of the write-ups and looked at whether there were approaches that were associated with better performance. We had one group that worked on reproducing all of the solutions from the code. And then we had a external group that worked on ensemble modeling. So the most interesting thing that came out of the working group from my perspective is that there was actually very little overlap in the sets of predictors that each of the groups came up with. There was no gene that was used by all of the same, by all of the models, and the overlap between models, predictors by the models drops off pretty quickly. And we also saw that none of the individual sets showed any sort of biological enrichment, which sort of implies that each model is picking one or few genes to represent a whole biological mechanism. But when we aggregate those across models, we do see that there is biological enrichment both at time 0 and time 24. And that's not picked up by any of the individual models. We saw that, so that sort of implies that each model is picking up different genes to represent the same biological functions. And so here we saw that heme metabolism and erythroid differentiation were the most associated with infection at time 0. We also see that at time 24. At time 24, we see much more immune response, interferon gamma, that aren't complement, that aren't showing up at time 0. So that, and then when we combine across all of the time points, we see that heme is by far the most enriched pathway associated with symptoms. And then what we see associated with lack of symptoms are sort of homeostatic processes like oxidative phosphorylation and MYC targets. And in the interest of time, I'm going to skip through the last of these. But just in summary, challenges offer a novel form of collaboration to solve important problems and benchmark solutions. In the respiratory viral challenge, we found that response to virus can be predicted even prior to exposure. And we were, based on these collaborative analyses, we were able to identify biological pathways that are driving these predictions. We also, I didn't talk about this, we also found that some subjects were consistently difficult to predict and some were consistently easy to predict. But, more information or more study is needed to see what drives that lack of ability to predict. This is all, challenges in general are driven by our team at SAGE. And then just to acknowledge the viral Dream Challenge group, we had a group of organizers from SAGE, from DARPA, and from Duke. And then, of course, we had a large community phase. So with the people who drove the bulk of the analyses highlighted in bold there. So with that, I'm happy to take questions.
Okay, thank you very much for an extremely interesting talk that's highly relevant to the things we've been talking about. If it's okay, I think we will follow our usual protocol and continue to our next talk, which will be by Julia Arciero on predicting survival with a mathematical model of acute inflammation. And then we'll have an opportunity to discuss freely with both of our speakers. Julia, are you ready for presentation?
Well, thank you again for this invitation. I'm excited to be here. I see some parallels between the first speaker and what I'm going to talk about today in terms of understanding disease at early time points. So maybe that'll be a topic for discussion later. But yes, I'm a professor at IUPUI, so in Indianapolis, and I do a wide range of work, a lot of blood flow modeling, some transplant rejection modeling, and I don't do a ton on sepsis, but I thought this might be an interesting project to share with you. Some collaborators of mine at the University of Pittsburgh presented a data set to me, and I'm very interested in inflammation and immune response. And so they had some data on sepsis. And so we looked to see if we could use some modeling to understand the types of data sets they were identifying. So kind of what is sepsis, or how do we understand sepsis? So the idea is anytime you have an infection or an outside, foreign agent, your body is working very hard to mount an immune response to defeat that pathogen. And the problem is sometimes that immune response is overactive. So basically, instead of maintaining kind of a local immune response to defeat the insult, you get this really systemic inflammatory response that starts to be overwhelming and really causes damage to the host tissue and organs and can lead to organ dysfunction. And this is what happens in sepsis. And this has been studied from many perspectives for many, many years. And one question that often arises is, why do certain hosts mount this overwhelming inflammatory response? And really these patients succumb to sepsis, as opposed to other cases might have a similar infection and yet they don't have this overwhelming response of the immune system. And so really understanding this dichotomy would be a great way to intervene and provide treatment for these patients to prevent septic death. So one of our goals is, are there mathematical methods or modeling techniques we could use to identify when you would have a septic trajectory? So given some sort of disease, can we understand early on what might lead to sepsis in some patients and not in others, so that you could intervene accordingly. Now, I'm not saying we've solved that problem, because we certainly have not, but we're providing here a model that it's a very basic model, but just that can start to look at some of these questions and try to understand some different mechanisms that could be contributing. And so here is the data set, two data sets actually, that my collaborators provided to me and our team in their E. coli experiment in rats. So basically on the left side, I've labeled a calibrating data set. And so this is where they had a group of 31 rats with they were injecting with increasing doses of E. coli, and this was a peritoneal experiment. And so here are the color coordinated different doses. So between 1.28 times 10 to the 8th bacteria up to 5. So they kind of doubled and then doubled again, basically. And they were measuring bacterial levels over time course. And they, for the highest level of the bacteria that was implanted, this was implanted via a fibrin clot. They were taking much more short time frame because they knew that the rats would die quicker for the higher dose. So you see kind of a larger clustering of data points early on for the highest amount of bacterial being implanted to these rats. And then, most of them are dead within 24 to, 35 hours. And then the darker blue is the medium dose, and they survive longer. And then the light blue survive the longest. And in fact, basically the rats were sacrificed at 96 hours, even if they would have survived longer. Most of the time they were pretty unhealthy, but there were cases where they were not and would have survived longer. So they could have been considered healthy rats, but the protocol was such that they were sacrificed at 96 hours. So this was the data we used to calibrate and I'll mention this throughout, some of the data or some of the parameters in our model. And then the second data set they provided us with is on the right-hand side, called the validating data set, where what's being plotted is the injected bacterial dose on the vertical axis. And then so this was I think this was about 20 some rats in this data set. It's a different cohort of rats with again, similar order of magnitude of bacteria, but different, varying doses. And then they observed only at 4 time points. So basically at 24 hours, they observed how many rats died. And then, so this is observed mortality time on the horizontal axis. So basically, if a rat died anywhere between 0 and 24 hours, it was characterized as a 24-hour mortality time. And so forth, 48, 90, 72, 96. So in little blocks of definitions for their observed mortality time. And so we use this to compare and understand how our model was doing against the different data sets based on how we had calibrated it. And a couple of things really struck us by this data set, and most importantly is the fact that you could inject the same dose of bacteria and have a very wide range in survivability time. So I've highlighted 2 specific rats here, like rat 11 and rat number 20, who have nearly identical initial bacterial loads, and yet one died at 48 or sometime between 24 and 48, and the other one died by 96 hours. And so the question becomes, from a modeling perspective, can we understand why this might be? And then, we performed some statistical analyses, and I should mention that on average, there was not statistical significance within the 48 to 96 group. So we ended up dividing observed mortality time as being less than 24 hours and greater than 24 hours. So I'll show that to you later. Okay, so given that data set, which is what they provided me and I worked with another faculty member, and we also used this as an REU project. So I had several REU students working on this, I think 3 students. We have some questions that were motivated by this data, namely, number 1, why does the mortality time differ dramatically between rats given these identical bacterial doses? So that's one question that my students sought out to answer. And another important question in the area of sepsis is, is early data on the pathogen and on the host response sufficient to predict whether you're going to have a health or disease outcome? That to me is a very important question. And can we answer that? And what can modeling tell us? So those are some of the questions that motivate this work. And ultimately, we decided to combine some inflammatory response models that the team, so I actually worked at Pittsburgh as a postdoc. And so I had modeled necrotizing enterocolitis, and then Angela Reynolds had modeled just kind of basic viral, inflammatory responses to viruses or bacteria. So we ended up using kind of some of this history of our modeling work with the team over in Pittsburgh to create a similar model, kind of combining those two studies, and to be real basic. So we're not modeling tons and tons of cytokines and all of these other things. We're really saying, here's a bacteria. So in this case, it would be E. coli. You have some dosing, some external amount, right? Because they're implanting these fibrin clots. And then in response to the bacteria, you mount a pro-inflammatory response. So we're lumping a lot of things into that category. And then inflammatory response kind of feeds back on itself. It also triggers anti-inflammatories to the scene, and it also causes damage. So we are referring damage. I mean, you can think of them as damps, but you can also think of it as like overall health of the system, but basically damage to kind of the host tissue. So basically it's causing damage. So pro-inflammatory response is doing a lot here. Anti-inflammatory response is trying to keep things at bay, right? So it's trying to, it'll inhibit that pro-inflammatory response. It will inhibit damage. But we also see with damage, you have this self-sustained loop of pro-inflammatory response and then more damage, et cetera. So it continues to feed back. So this is how sepsis can occur and can be very certainly dangerous for the host. So that's the basic model. I'll share with you it's a differential equation model. Again, like I say, pretty simplistic, but we did that on purpose just to understand relationships between these populations and to see what are some of the factors that are most important that can help us to answer some of our motivating questions. So here's our bacteria equation where you have this external dosing function. We tried different forms of it, but ultimately it wasn't that sensitive to it. And we chose this exponentially decaying function where you implant it and then you have kind of a decay. Some logistic growth of the bacteria, and I circled K1 as a pathogen growth rate, since that will be one of the parameters we consider throughout. Then we assume a local immune response. So this is, of course, the negative term, because you have kind of this local or innate immune response with some parameter K2 that indicates its strength. And that is going to limit bacteria as well as the pro-inflammatories. So population M meets bacteria B and eliminates bacteria. And this is inhibited, or, again, that inhibitory aspect of the anti-inflammatories we see in the denominator. And the pro-inflammatories, of course, as you saw from the schematic, are activated according to themselves, to the presence of bacteria and to damage, and it's inhibited by anti-inflammatory response. But this new one parameter, again, is an important parameter which helps to define, or it characterizes the activation rate of the pro-inflammatories. So that's another one that we'll look at. And then we have some decay term. Anti-inflammatories are assumed to be at some background level. So, and then we would say they're triggered in the response of pro-inflammatories and damage and reduced by anti-inflammatories. So again, these equations follow very directly from the modeling schematic I gave you with some decay. And then the damage might look a little interesting. So the most important term, so we have a repair term, but basically we have this max function, which is saying, how is damage generated? Well, if the amount of anti-inflammatory response is greater than some threshold, then it's based on that difference. But if it's less than some threshold value, then we assume it's zero. So it's basically the max of this term and zero. So it's to say that your body can handle some degree of inflammation before it actually mounts a ton of damage to your system. And that's kind of how we incorporated that into the model. This can be done from different perspectives, but it was one way to do it. Okay, so I'd like to show you some of the results we have from this model. Notice the log scale here on the bacterial levels in case it looks funny. But so really we can have three outcomes that the model predicts. So we can predict the case of health, asepsis, or aseptic death, or septic death. And what do we mean by those three categories? So a system will be healthy if the bacteria are cleared and the damage goes back to, baseline or low level. So you need clearance of bacteria and damage. You can't be damaged. You don't want any sustained damage. So then the person or the rat would be healthy. Aseptic death means that they've cleared the bacteria. So the inflammatory response did its job, but it did it too well because it caused a bunch of damage to your tissue. And so damage levels remain elevated, as we can see in the dark blue curve. So see that, 10 to the minus 2, we see things going to zero in the bacteria levels, I should say, going to zero in both the healthy and aseptic case here. But in the health, damage is zero, but in aseptic, we see that damage is elevated. And then septic death corresponds to the case where you would have bacterial levels remain elevated. So you haven't cleared the infection and you're severely, damaged, like high levels of damage. And so what we showed was for this particular model, where I should mention, I guess I didn't say that while I was looking at the equations, but we had, because I mentioned it was based on a model by myself and Angela Reynolds, two different models, we actually had a lot of those parameters from previous models. And so we used many of them because it was an inflammation model. And then we did a sensitivity analysis and determined, and using that calibrating data set, we fit 7 different parameters in the model to that data set that we were provided for those rats. And so this is the outcome where in this slide, I'm varying K1. So K1 was the pathogen growth rate. And you'll see in a future slide that we vary other types of parameters, but this is just one that says, okay, if a characteristic of the pathogen were slightly different, then we can actually get vastly different outcomes. So varying K1 between 1.2 and 1.4 actually leads to anywhere between health and sepsis. And so that was one way for us to demonstrate that the model can actually lead to many different outcomes despite the same initial load of bacteria. Okay. So then we wanted to set what we were calling epsilon critical. So epsilon critical is this critical level of damage where we're basically saying, okay, the time of death, so I'm calling it T sub C, is what the model predicts the death is based on the value of damage reaching some critical level. So we could vary the critical level and see how accurate our model predictions would be against that, against our data set from the rats. And we see that we are more than 95% accurate for an epsilon critical, somewhere around 55 or so. And so we were using this to, and ultimately we do pretty well in general. I mean, for a large range of critical damage levels, we're above 80%, but we chose epsilon critical to be a value, I think it was about 55, to account for the fact that that meant our model had an accuracy of like 96%. Okay, so then we were very interested with using that other data set where they had all these different initial bacterial loads to see what the model would predict. And so we tried all of those as different as initial conditions. And what I'm plotting here in blue, the dark blue, is a rat that would have an observed mortality time less than 24 hours. So kind of like that first cohort. And then remember that other cohort was the last 3 sets of 48, 72, 96. So basically observed mortality greater than 24 is in red. And so I'm plotting, again, bacteria, pro-inflammatory, anti-inflammatory, and damage with time. And we see a very clear separation between aseptic and septic outcomes, right? Because we see here damage is staying elevated for this particular set of data point, or these particular initial conditions. But what's not the case is that the red and blue don't necessarily separate along with the septic and aseptic, right? So it's not like there's a clear distinction based on their observed mortality time, whether they become septic or aseptic. Moreover, we kind of see a clustering at early time points where you can't necessarily distinguish things like bacterial levels or inflammatory cytokines, things like that. So there's quite a clustering without spread early on. Okay. And then we wanted, if you remember on that validating set, I showed you 2 different rats that had nearly identical initial bacterial loads. And we wanted to understand, can the model predict scenarios where those rats indeed have those different mortality times, despite the fact that they had the same initial condition or initial load. So here's one example, and I'm going to show you a whole parameter investigation or bifurcation set in a sense for different parameters. But if we were only varying that pathogen growth rate K1, then we see indeed that we can, here's epsilon critical, here's that damage threshold. So rat 20 remained healthy because damage was low. So this was for a pathogen growth rate of 1.2 per hour, and bacteria are cleared. But then if you increase K1 just slightly to 1.27, there it crosses the threshold at the observed mortality time from the data set, basically. And so indeed, a small change in that particular pathogen growth rate allows the model to predict the disparate outcomes that were observed in the validating data set. And so, like I was saying, we could do that not just for pathogen growth rate, because one might argue maybe pathogen growth rate is not the most relevant parameter to vary in a sepsis model, especially if you're using E. coli. And so I've done this for lots of different parameters. So this was on the left is the pathogen growth rate, but now I could also vary the strength of the local immune response, or I could vary the pro-inflammatory activation rate. Again, with the thought that people have, their ability to mount an inflammatory response is definitely varied. We see that, today in the case of the pandemic, we see people's responses to be very different. And so it's a very patient-specific aspect of the immune response. And so these were 3 of the main parameters we looked at. And I'm always plotting B source. So by B source, I mean the initial bacterial load on the vertical axis. And then I'm allowing the parameters, these 3 different parameters that I've chosen, to vary on the horizontal axis. And I'm mapping out for you what the model predicts in terms of either healthy recovery, aseptic, or septic death. And I've drawn dashed lines at those original, in the validating data set, those original 3 levels of bacterial insult of E. coli, just to give you a sense of where those fall, given the range of parameter values. And then the 3 diamonds correspond to that first data set, or that first model response I showed you, where we varied K1 and showed you could have health or aseptic death or septic death for the same initial bacterial load, just 3 different values of pathogen. And then the 2 circles correspond to my 2 rats, rat 11 and rat 20, that again could have been healthy or aseptic, again, given nearly identical bacterial loads here. So we can see, certainly as you increase the bacterial load, so if you're up at the very highest one, the highest dashed line, the model predicts you would only have an aseptic or septic outcome, regardless of parameter value. And now I probably should have drawn a vertical line to show you kind of the fitted or the typical value we use. I think it's usually, somewhere around 1.2 somewhere in the middle. I think we tried to make it so that the plot was more or less centered around the main one. And so it kind of intersects all three curves, is what ends up happening, or all three regions. But so again, kind of as you would expect, if you have a low immune response, then you are certainly more likely to become septic. And, you know, if you have a high inflammatory activation rate, you're more likely to become aseptic. But it's just a way to map out and explain to someone from a more mechanistic perspective, look at how sensitive the results can be. I mean, look at this region here. Look how sensitive the results can be to these kind of inherent parameters that are defining specific mechanisms of action of your host response. So we thought that this was a pretty interesting way to showcase that study. And so we'll leave some time for questions. I'll just conclude with saying, you know, we showed pretty serious variability in outcome as you vary parameters that affect pathogen growth rate, strength of the immune response, or activation of pro-inflammatories. And so we were able to show, using our model, the difference in this case experimental, but basically the idea is it would relate to clinical variability, that really small differences either in an initial condition or in these parameters can really lead to different outcomes. And I guess another point is that while potentially you could do this from a fully statistical standpoint, we wouldn't really be able to understand some of the mechanisms or explanations of these governing components without having kind of a corresponding model to go along with it. The model predictions also showed that early time points at like 8 hours were not able to be predictive of the actual outcome. And ultimately, we would say, regardless of what's causing the inflammation, whether it might be a trauma or cancer or some infection, a virus versus bacteria, whatever the case may be, there are published studies that show all these dichotomies of whether someone becomes septic or not, and whether, you know, they're in the hospital for weeks or having issues as opposed to recovering without a problem. And so we believe that kind of a combination of both the mathematical mechanistic modeling and statistics will become very important to understanding this problem. So I would like to acknowledge Yoram Vodovots and Rami Namas from Pittsburgh. So Rami did a lot of the experiments, and Yoram is my collaborator in immunology. And Jared Barber worked on this at IUPUI from math, as well as some of our RU students who are now, 2 of the 3 are now in graduate school. And we thank NSF, NINA, and NIH for support. Thank you.
Okay, thanks to both of our speakers. Actually, the talks, as you say, went together very well. I thought that was a nice combination. So there's a time for some questions. I know we have a somewhat smaller group today, and some of our people have to leave at 4, but I think there's an opportunity to ask some questions. I have some questions for both of you, but I'll leave it to others just to go first, if you like.
Well, thanks both for the talks. Were amazing, both on different sides. I have one for Solly, actually. It's really interesting to me. Well, all of what you said, but there was a part when you had this problem when you had to integrate some data that you couldn't show to the public, and then you use the docking strategy to bring some model. Well, in that case, it was bringing it beyond the hood, so it couldn't be seen outside. But I think the docking strategy can be used for several things. So, for example, to combine different models, etc. And I had another question: is okay, what is the kind of question that you take? So, as an entry point for someone that wants to engage, is a biological question, or is you propose challenge and people plug in, or things like that? So
So yeah, to your first point, we've started using Docker more and more regularly, even when we can share the data, just because it helps with reproducibility, because that's one of our one of our goals in challenges. When we ask people to submit code, it's only some of the portion of the time is it actually reproducible, and so Docker helps us at least make sure that the models are runnable. Whether or not you can retrain them is often a different question, but yeah, so it's a great tool, and we love it. In terms of starting a challenge, I think there are kind of two approaches, or multiple approaches, really, depending on what you're interested in. You can come with a biological question. You can come with a methodological question. The tumor heterogeneity, the somatic mutation challenges were an example where that was purely methodological. The field needed to understand what tools were best for that problem. Sometimes you come with a biological question. Sometimes you have a really cool data set, and then you figure out what questions you could ask with it. So I think those are three possible entry points.
Thank you.
Other questions for our speakers? I had a question for Julia. In terms of your workflow, if I understood, you have data sets for individual animals, and then you're fitting 7 of your model parameters to fit to each individual animal data set, and that's what you're calling animal 1, animal 17, and animal. How did you do it?
No, so that initial calibrating data set, which was 31 rats, we had time point data for bacteria levels. And so we were fitting the 7 parameters, the basic model, to give a best fit. Right. I guess I should have shown that figure, actually. I could have shown the fit to that whole spread of data.
So then what was the difference between your simulated rat 17 and your simulated rat 11?
So basically they measured a set of rats, and then they measured a different set of rats, and they had different doses, basically, not necessarily the same doses. So we basically took the initial condition of the initial bacterial load, right, that they put into to each rat, and so we just simulated what happened to each of those.
But you only had one parameter set, and then you did variation around that parameter set.
Right. Mm-hmm.
Okay. I thought maybe you fit a bunch of different. You generated a synthetic population where you had rat number 11 that was
right. No, we did not do a fit for every rat. 20, no.
Okay. Okay, so that was what I didn't understand. And did you use a single best fit parameter set, or did you use a cloud of parameters?
So this took us a little while, because I will say that this data set was not ideal, so we could use a bit more data. So we did a lot of different tests on this, and we did basically a sensitivity analysis and found which ones were most sensitive, and then we fit to like log normal data versus regular, and so we ended up having to do log normal. And so we did. Does that answer your question? So we did a variety of tests to see what would be most successful. And again, ultimately, if we had a bit more, because not only the clump, they only had so many data points as well. And so like they had clustered them more early for like the high dose, but then we only had so many data points for time. And so I think if we had a bit more data sets and more rats, honestly, it could have been a much better fit. I think the mean squared error ended up being like a 0.7.
Yeah. But I guess, no, I was asking was, when you did your prediction and your time and your forward sensitivity analysis, did you do that around a single consensus data set of parameters? You had the value of this parameter is this, the value for this parameter is this, or did you have an ensemble of parameter sets that were approximately equally good that were within your AIC or whatever acceptance criterion you use, so that you were doing a cluster of models?
So I have to be honest, Jared did this. So if I remember what he did he did the statistical work basically. Well, I know that we always set the certain number initially come from the other model. And then I think that he tried to do a cluster based on which would give the best error result, but I'd have to double check with him.
It looked like you were using a single best fit set of parameters, but I wasn't sure. It's a lot harder when you have another cloud of parameter sets.
Do you mean when you actually started? Well, when we started to run the model, we used a single parameter set. Maybe I'm not understanding what you're asking. That's. Oh, yes, absolutely. We did a single set. Yes.
So you didn't have, because you're overfitting, so you could imagine that you accepted a whole pile of parameter sets and then you did some kind of particle swarm method forward to look at the ensemble of prediction.
Right. Yeah, we didn't do that.
No, that's fine. I just wanted to be clear.
I see what you're asking now. Mm-hmm.
Partly because I was thinking in the context of the previous talk.
Right.
And asking the question, and I think this would be something for Solly, which is, we're usually a slightly bigger community than today. The good news is that this is recorded, so a lot of people watch it offline, even if they can't watch it at the time of the presentation. So you'll have a bigger impact than maybe the number of people who showed up just for this. But I wanted to come back to the question of organizing these challenges. It's always seemed very appealing to do a challenge. And we could think this idea of trying to develop immune digital twins, there could be many possible challenges. There's also the question of simply doing model replication itself. In the sorts of modeling methodologies that we use, the ODE models, or for that matter, some of these neural network models, there's a standard set of methodologies, and therefore replication or reproduction, at least in principle, is possible. When you go to agent-based models of the kind that I developed, that Lorenzo does, that Paul Macklin does, that Reinhardt does, there isn't any kind of shared methodology or language for describing these problems. And so we found repeatedly that replication, running the same model, is already hard. As you pointed out, just because the code's published, you can't necessarily get it to run. But reproduction becomes really problematic. Can you actually, if we have a simulation that gives you certain results, can somebody else, based on the way we published that and our description of it, can they reproduce it using a different methodology? And with the exception of the Oxford Chase group, almost no one has attempted to do this in the context of multicellular agent-based modeling. Now, that may not be a sufficiently exciting challenge, but I was wondering, first question would be a little bit about how these kinds of problems might differ, if at all, if we were talking about the kind of mechanistic model that Julia was talking about, rather than the more data-driven ML approaches, which it sounds like are the majority of the kinds of challenge you've done. I suspect the answer is not very different, but I wanted to understand the specific issues that might come up in that case.
Yeah, I mean, I think if your metric is, if you have a metric against, against which you're measuring, Julia showed her accuracy over a variety of parameters, I believe, if I understood correctly. So, if you have an accuracy metric that you're interested in, you can compare models, making sure that you're comparing on the same metric, the same data set, et cetera. So from that perspective, I don't don't see a difference. You know, the later portion where she was looking at the model spaces and the actual predictions, you know, that I guess would differ from model to model, but you know, in terms of comparing models and comparing model accuracy, I don't see a difference at all.
That's very helpful. I think that's something that's interesting to think about. The other thing, which is more of the community side of it, is do you see these kinds? You talked about community building, and one of the challenges, of course, one of the questions I had, and then you showed a slide that showed it, was how long does the challenge last? And the one you showed was 4 months.
Yeah, typically an open phase will be 3 to 6 months, depending on the complexity of the data and the complexity of the challenge. But you know, that can vary. Yeah, one thing we've wanted to do a better job of is aligning better to academic calendars so that we can leverage, you know, students who are, you know, leverage classroom settings and things like that as well. So I think that's also something that we want to take more into consideration as we're designing challenges.
I mean, it sounds like in the challenges that you've had, you don't face the one we might face here, which is that our community is not very big. And so there's a question of how many people would show up if we did a challenge. I think somebody like, I mean, the example Julia gave actually could be a nice challenge in a sense. Maybe it's a little bit, the data set might not be sufficiently discriminatory, but conceptually it's a good example. Julia showed a very nice model based on her work and Angela's work that was this very simplified model of the damage and regulation of immune response. There are people who could come in with a more elaborate model with more parameters. Somebody might come in with a spatial model, try to do the same thing. All of those could be trying to predict those time series. There is no consensus really on what level of granularity is needed to model inflammation and sepsis. And so I think that could be an excellent challenge problem. As Julia said, perhaps this particular case, the data set's not sufficiently rich that you'd want to use it as a challenge problem. But if one had, last week we had Ben Tanover from NYU talking about long COVID. And he has incredibly detailed data sets that are publicly available, both time series and genomic. His hypothesis about long COVID is a chronic inflammatory state hypothesis. And so in a sense, if one could take a set of the kinds of models of various types that Julia showed in that space and use those against his data sets, that could be a very, very exciting challenge. Long COVID is a big deal, of course. The news today, there was an editorial about what they called moderate or medium COVID, which is you do get clearance, but the clearance still sorry, I should be careful about clearance as a word. Resolution of these long-term symptoms, but not right away. 3 months, 4 months, 5 months. So there's this intermediate state of persistent inflammatory or damaged state, which then eventually clears. And so there potentially are additional outcomes beyond the 3 examples that Julia gave in her bacterial case. Seems to me that could be really an interesting challenge data set to try to work with. And my guess, just to prejudice, because like Julia, I'm a mechanistic modeler, is that a pure data inference method wouldn't work on that prediction. But it might. And it would be very interesting to compete mechanistic, hybrid, and pure ML methods against each other on a data set.
Well, our community is very ML based. So, if you had a way to advertise to the mechanistic modelers, and we could certainly bring both to the table and see.
I mean, as I say, unfortunately, I apologize for, I don't know what happened today. We usually have 25 people in the room. As I say, a lot of people do watch over on the Zoom. I mean, on the YouTube, and also this is spring break for a lot of universities, and there are.
I was gonna say it's spring break for us. Yeah.
There are a bunch of conferences going on, like APS at the moment, which has a lot of our
in Chicago. Yeah.
And so I think that the turnout today doesn't reflect the level of interest in either talk. From discussions, I know this idea of challenges and this idea of trying to build community through challenges is something that appeals to our community a lot. And so I think there would be, maybe not your 60,000 users, but you probably get 15 or 20 competitors. And the idea of using a class to target a project is something that's been talked about a lot. Cures, I think, is the educationalist term for those. Course, what is it called? Course. R is research. Where all the class does a project together, not necessarily all collaboratively, but the class all works on doing a model of something. Each person in the class works on a model of the same thing, either in teams or independently. And that, of course, follows the challenge structure very well.
One thing that I've seen around, and this brings to the machine learning or the computer vision side, which is one thing I'm interested, and where people were given a task which is easy to learn in terms of segment this or paint this image in classes. And this was kind of diffuse. It's online, everybody can participate, but then the project was to collect the data and sort out whether were meaningful or not, and then essentially use a diffuse intelligence to train a model, which is not a semi-supervised model is always a tricky task, no? Because if it's done by one person, it will bias it by its own nature. Yeah.
I know my collaborator, David Umalis, used commercial mechanical Turk outsourcing to do hand image segmentation to generate training data for his image segmentation algorithms. And somewhat to my surprise, it worked really well. Because even though the hand image segmentation wasn't always the most accurate, the failures were different depending on who did it. And so the ensemble data sets were actually really good for training his ML image segmentation. And it wasn't that expensive. We spent a few $1,000s to get 100,000 hand segmented images. I don't know if that's quite talking about what you were saying, Lorenzo, but I thought it was not irrelevant to that. I had one more question for Julia, which was a technical question, which we ran into a lot in our simulations, which is you have two kinds of data in your data set. You have time series data for the viral load, for example, and then you have time of death. And when you are doing fits where the data set stops at different times, that's not trivial to do. I mean, most loss functions, if we're talking ML, or just error functions or chi-squareds, don't like it if your data sets are truncated at different times. How do you handle the fact that you have time series where the duration of the time series varies between the data set? And how do you combine those sort of single point time outcome measurements with continuous time data? in your evaluation metrics?
Yeah. Let's see. So I think that so it was interesting. So so the data set I showed you, I think we showed every data point, and then they also had average data point at these different time series, but I'm pretty sure every data point was used. And so again, so my collaborator did the actual statistics on this. So I'm not totally sure if he ran. I know we ran into several problems again because of the sparsity of the data, but I didn't think that the fact that you had different time points necessarily caused a problem, and I don't think he had to do necessarily any special weighting. Although, again, there was a constraint function that constrained, because you had these 3 different levels of bacterial loads. So we had to constrain based on like the fact that you could not, basically the assumption was that we could not get a healthy outcome if you were the highest bacterial load. So that was something we did have to add in, which one could argue, is this valid or not? But ultimately we had to constrain based on bacterial load, like the level, because there were these 3 very different loads. But yeah, I think that more data and ultimately, along with what Solly's doing, it would be interesting to see if we had collections of data like cytokines in addition, something else to contribute to this would have, I think, made for a better fit and an easier time for us, and maybe more even better outcome predictions. I don't know if that answers your question, but I don't know. The actual number of data points along different time, like different stop and start, didn't seem to affect it that much as much as the load and whether or not you could get healthy or not.
Right. I guess I was just thinking in some of our simulations, we were doing tissue patch simulation. And if the infection is slow or contained, then the final number of uninfected cells is a useful metric.
Right.
But in a lot of the simulations, all the cells get infected, and then the metric is the time at which all the cells die. Mm-hmm. That's the most diagnostic. How do you deal with the fact that you have 2 different metrics? If all the cells die, the time at which all the cells die doesn't exist for the ones that don't have all the cells infected, and the number of uninfected cells at the end is a metric that doesn't exist for the other examples. Mm-hmm. And so then you have a problem that you don't have a single metric to describe all of your cases. And so in your case too, you have these branched. You have a dichotomy between the death outcome and the health outcome.
Right. And we had, so basically the damage, that critical damage level and where it intercepted it was kind of key.
Right. And so when you're dealing with those data sets, then when some subpopulation has one metric that's most discriminatory and some other subpopulation has another, coming up with a consistent metric of assessment may be tricky. Mm-hmm. Because ultimately, for the kinds of things Solly is talking about, it for your optimizer wants some number which is the goodness of fit,
right?
And that's got to be a single scalar number which is the goodness of fit. And so, what starts, I think Solly, you talked about that at the very beginning. When you're creative about that, you can game the system.